from the cell wall point of view, I think there's 4 areas where it's of interest to look at plant cell walls. It's first of all, this is essential in plant morphogenesis and responses to abiotic stresses. It's involved in biotic interactions, immunity. It's also involved, we should take it into account cell wall metabolism in calculating greenhouse gas emissions, but there must be emissions, and I will talk about the bioeconomy. So morphogenesis, abiotic stress. So lignification, I don't need to talk about that because Wout did that very brilliantly this morning. Another application is heavy metal tolerance. I think maybe Wolfgang will mention that or not tomorrow.
At a slide.
So that is work of Wolfgang, who actually showed that there is a natural variation in pectin metal esterification. I will talk a bit more about that later. That is involved in natural variation in plant zinc tolerance. There was a recent paper of José Dinini lab where he showed that hydropatterning, it's a important trait in crops, because you see it on the scheme here that if you put a maize root in the presence of water and on the other side is air-filled space, you see that the roots tend to move towards the water, and they studied this pathway that is regulating this process. And one essential component is this here, which is a arabinogalactan protein that seems to act as a sensor for water availability. So it's associated with the cell wall. So biotic stress, a lot of QTL data and natural variation that shows that cell walls play a role in disease resistance. You have in maize, you have several cases also in different maize QTLs. Also, there's also in again in maize. I think there's also some in wheat. So these are WACs. These are wall-associated kinases that somehow recognize cell wall components. Wout knows well about this. Probably that you have also in the phenolic pathway. You have also QTLs that provide resistance against pathogens, and this is work from our group that showed that in the botrytis Arabidopsis interaction, there's oligogalacturonides that play a role in the interaction. And then greenhouse gas emissions, and Kalina, for those who saw her talk, already mentioned this, that it's a recent paper that showed that actually the photosynthetic C1 pathway, so it's a phosphorylated pathway of serine biosynthesis, that it's a dedicated pathway that's going to produce the precursor of methyl groups in polymers, which is here the adomet here, which is the donor, which is specifically produced from this pathway that is going to produce the methyl groups among others, the pectins, which is a major component, methylated component. And these pectins are going to be secreted and demethylated by pectin methyl esterase. I will talk a little bit more about that. And it releases methanol. And so what is interesting is that during growth, there's a very important pectin turnover. So there's not all the pectin is going to be used to make cell wall material, but it's like being produced during the assembly of the cell wall. You can easily see that here in a root hair here is all the stuff that you see here that's endocytosed is actually demethylated pectin, which is the pectin that is like turned over during the growth process. So there's a very important turnover, and that releases a lot of methanol, but that can be converted in methane. So it's something that needs to be taken into account in measuring greenhouse gas emissions, actually, which is associated with the growth process. And another in relation to greenhouse gas emissions, and Claire Chenu, who gave a talk last week, pointed this out, that the root mucilage is actually important in creating, rhizome deposits, which is going to influence the ability of the soil to accumulate carbon. And there's a few examples that show that the root mucilage is actually going to play a role in this, and that there is some natural variation also in the ability of the mucilage, the amount of mucilage that produced, but also the ability of the mucilage to contribute to these root deposits, the rhizome deposits. Okay, so the bioeconomy. So you use biomass. So the lignocellulosic biomass is an important source of carbon if you want to replace the fossil carbon, and you're going to use it at many different length scales. So you have example of the roof of the Notre Dame, which is wood that you can use, but then you can also fragment the wood and use fragments. That's a project that we had to produce concrete from miscanthus fragments. You can also make paper, of course, which is a more enriched cellulose, and you can make all kinds of objects with regenerated cellulose. And of course, you can use lignin, and Stephanie gave examples of that. And also the hemicellulose can be used as a material and to make materials. And of course, you can degrade everything and then make ferment and then make biofuel. So you have this, you can use it at many different length scales, the biomass. And that, of course, has different quality requirements depending on what you're going to do with it. So that's it's one of the complexity of using biomass or lignocellulose biomass for different end uses. So the feedstocks, you can use agricultural residues, straw, wheat bran, which poses a number of, even if you use those co-products, it poses a number of problems because they are used for other things as well. So there's always a competition with other end users. Wood and forest residues you can, and then you have also dedicated crops. So Wout already mentioned poplar that can be used, but also miscanthus, which is a perennial grass that that can be used. So concerning the choice of the biomass depends very much on local conditions. You don't transport it very far because the density is very low, so it becomes very expensive when you want to bring it over longer distances. And so it really depends on very local conditions what you can use. So I will talk a little bit about this dedicated crops. Can be a solution in some cases, has a number of advantages, the yield, the environmental footprint. Give an example, miscanthus, and then you can reduce the competition if you put it on marginal lands or use it as an intermediate crop. Here, this is sorghum, for instance, that you can grow in between two harvests. This is 2 months, so you can get quite some biomass in a very short period. These are sorghums that have very high biomass, but we talked about robustness the other day. I mean, these are not very robust. When there's a little bit of wind, or they tend to lodge very easily. Miscanthus is a perennial grass. So why would you bother about perennial grasses? It doesn't move any biome. So what is interesting with these perennial grasses is that they have a rhizome which is going to stay in the soil. So the plant is going to have a growth season during the summer. It will senesce during the winter, and you can harvest it in March, April. And so what is interesting is during the senescence period, you're going to so this is the rhizome, this nitrogen content. You see the nitrogen content goes down during the growing season, goes up in the aerial part. And then during the senescence period, it's recovering the nitrogen into the rhizome and it's going out of the aerial part. So it's a very nice system to export carbon from a field without having too much impact on the minerals, and so it's also the case for other minerals. So so that's an advantage of these perennial grasses. They have other advantages. So this nutrient recycling, there's a very favorable energy balance because you have very few entrants, but also very few interventions. You only need to harvest it once a year. And the yield is high. So the energy balance what you export from compared to what you put in the field, it's very high compared to other crops. You don't do tilling, so it's very good for the soil. We discussed all these things already. Carbon storage also. Water management, which is interesting. They have a deep rooting system, but they have an early growth season. So compared to maize, which grows mainly during the summer, so you get also the advantage that you grow during when there is still rainfall, etc. And then it's compatible with marginal land. So in France, it now has become eligible for the ecological focus areas, which is a water catchment area. So where you cannot do agriculture because it's like people want to avoid contaminating the drinking water with minerals, nitrate, etc. So in these zones we can grow these miscanthus, for instance. Has a low water content when you export it from the field, because you harvest it when it's really senesced completely. And France, it's still marginal. It's 11,000 hectares, but it's increasing every year. It's 14% the last 10 years every year. So the farmers kind of like it. The problem is that it's mostly clonally propagated, the plants that are grown, and that's of course a problem because it's almost a single clone that is everywhere grown, so we need to diversify, and it has a high establishment cost. So the field of a hectare is about 3,000 euros to install, which is a high threshold for a farmer to grow those things. So that's a little bit about the crops. We can discuss that maybe this afternoon in more detail. So I will talk now a little bit about cell wall research challenges. With a couple of scientists, among which was Wout also, that we wrote a paper where the main questions in plant cell wall research are listed. And one of the important things is to understand the structure-function relation of cell wall polymers. So the plant cell walls are very complex. You have a very complex set of polysaccharides. You have all the hemicelluloses, you have the pectins, you have, of course, the cellulose, shown here is a linear polymer of glucose, beta-1,4 link. And you have also very complex, like the AGPs that are attached to proteins and things like that. So you have it's a very complex combination of polysaccharides. And the question is, why do we have all this diversity of polysaccharides in the cell? What is the structure-function relationship in vivo, but also when you're going to use it for technological applications? And so that's it's really an important question that's still in the field. I will give a few examples. Hemicellulose is the work of the group of Paul Dupré, who showed that actually the xylans, which are main polymer in the secondary cell wall, they have different domains. And you see here, so they have what he calls the major domain, minor domain. And they are different by the fact that the substitutions have a periodic nature in such a way that when the polymer is like exposed to cellulose, it's the substitutions on one side, so that it can easily interact with cellulose. But then you have also parts of the polymer that are randomly organized, and that is actually preventing the association with cellulose. And this is going to play a role in the lignification of the cell wall. So it's so the distribution pattern of these substitutions is critical for the functional properties of these polymers. I think it's a nice example of the structure function, or some understanding of structure function relationship. There's an application of that is the group of Mathieu Rimond in our institute. They used actually a mutant that has an altered distribution pattern. This is arabinoxylan. These are arabinoses that have also the same distribution patterns. And if you modify that pattern, actually you can produce a maize that has a much less lignification, as you can see here with this staining for lignin, which is pink when there is lignin and blue in polysaccharides. So it has an impact on the lignification by the way that the substitution patterns are organized. So another example is with when you're going to, you can use the xylan, the arabinoxylan, you can purify it, and you can make films of it here. These are like thermoplastic films that people are using now to make milk cartons, for instance. They put a layer of xylan film in it because that's some interesting properties for gas exchange and things like that. And what is interesting is that the tensile properties or the ductile properties of these films are very dependent on the substitution pattern of these arabinoses. And so you get a very ductile material when you have more arabinoses, for instance, on this polymer. So which is like a technical property that is determined by also the structure of this xylan, so which is an interesting application. But it's not always so simple, because you can wonder maybe they all have a function, these different substitution patterns. It's not always so a bit surprising. For instance, here, the xyloglucan, they have also very characteristic substitution patterns here. I won't go into detail here. But what they show in this paper is that when you remove this is a galactose. When you remove this galactose, actually this is in a normal plant here. This is a cell. This is the representation of the cell wall with the xyloglucan. And you see that when you remove the galactose, actually the xyloglucan becomes totally insoluble and aggregates inside the cell. So you have this galactose is there to prevent it from aggregating during the process of secretion. And they could actually replace it with another sugar. They had it replaced with a fucose, right? So it's with a specific enzyme. They added a fucose instead of a galactose, and then it became soluble again. So that's if you try to understand the structure function of a polymer, you need to take into account also how it is produced, and that there's some constraints during this process. So something about pectins, that some work from our lab is that pectins, so the homogalactronan is a linear polymer of galactronic acid, which is this charged sugar, and it's produced as in a methyl esterified form here when it's secreted into the cell wall. And then, as I mentioned before, this methyl group comes from this particular C1 pathway, but then you have an uncharged polymer. So then you have enzymes that will remove the pectin methyl esterases, will remove these methyl groups, and that it can do in different ways depending on the enzyme, depending on the conditions, can be random or blockwise. And they change totally the physical chemical properties of the polymer, because you start adding like negative charges in it. And so another thing is that this PME activity is also regulated by inhibitors in the plant. So it's a highly regulated process, and it's important because is actually central in the control of plant growth. There's an example here of Alexei in our group who studied that by manipulating the metal esterification level of the homogalactronans in vivo. So this is a wild type plant. You see here, this is a meristem here. They make primordia, they grow out, they make flowers. There's a flower meristem. When you produce more PME, so that you actually demethylate the pectin more than it's normally the case, you actually get new primordia. They actually, the meristem grows bigger and they make new primordia. So it affects totally the development of your plant. And if you prevented this demethylation, you make a naked meristem. So you can see it here. They don't make primordia anymore. So it's you have like a link between the physical-chemical properties of this polymer and a developmental process, which is like very, very intriguing. And so we wondered if we have to do with what they call active soft matter, that you have the soft matter is the cell wall assembly that is like has its own regulation through the enzyme activity, and that engenders then properties at a higher scale. So to investigate that, the problem is that we have manipulated this, and we have this here, organ growth, but in between we don't know what's happening, and it's not a trivial question to understand the pathway. It looks, you say, okay, this is going to be properties are changed, so that is affecting the growth, but it's what's in between. It could also be that maybe there are some fragments produced that signal and then do this, and so it's really hard. We discussed it last week already, to these complex processes to dissect it. So one thing that we could see is, and Alexei did some atomic force microscopy, you could see that the cell wall elasticity increased when you demethylate the pectin. So that, okay, we say, oh, that looks exciting. So maybe the cell wall elasticity is going to control the growth then. But it might as well be this. It might be a side effect of the cell wall elasticity and there's something else happening. And so it's really hard to understand, to make this connection, because you bridge several length scales and to understand that. So what we decided is to look at a much more simple system, which is the root hair that I already mentioned. So where you can observe the growth process in real time, and then do things to, for instance, here we add an inhibitor of this PME activity. And so what you see is that this is a chymograph. So the cell is growing here. You see all kinds of other things. I won't talk about it here, but you see that the inhibition of the PME activity directly affects the growth. Is actually after 25 seconds, the growth slows down. And then you have after 50 seconds of feedback, where actually the pH increases at the cell surface here. So there's now you're looking at a process at a time scale of seconds, and so you can really start making causal relationships between the changes in your polymer and the effects on this macroscopic process. Okay. So another link between polysaccharide decorations and function is that it also determines the interaction with proteins. And that's a recent paper that we published, where we show that actually when you demethylate pectin, actually can bind this protein complex, which is made of a cell wall protein and a peptide, which is here shown in pink. That binds to this protein. This peptide is actually positively charged at the surface, and it's going to bind to the pectin, and it condenses the pectin. So you actually change out the water of your layer of pectin, and that's also happening in vivo. We could see that they actually this interaction with pectin induces the formation of a fibril network in the cell wall. So is actually driving the assembly of the cell wall. And what is also interesting is that this peptide, when it's not bound to the, when the free peptide is available, it actually binds to a receptor complex here that's going to regulate the secretion of the pectin, but also the demethyl esterification through the regulation of the pH, because the PME activity is actually has a high pH optimum. There is this seems to be a feedback mechanism to regulate its own assembly process, which is, but it's also related to the status of your polymer, the pectin polymer. So, and a last example where you have a link between polymer structure and function is the work of Mathilde. She's not here, but she together with Aline Foxer and Samantha Verhent in the lab, they showed that actually the interaction with botrytis, so and the botrytis pathogen in plants, botrytis produces a lot of pectinolytic enzymes. One of the enzymes is pectin lyase, that cleaves actually methylated homogalactronan, but only when it's acetylated. So it needs to have acetyl groups and methyl groups to be able to cleave it. And this it generates specific, this reaction of this enzyme produces a unsaturated end here that is actually going to play a role in the suppression of the defense reaction of the plant. And so I won't go into detail, but what they observed is actually the acetylation level of your pectin is actually dependent on the presence of nitrogen or the amount of nitrogen available. So you get more acetylated homogalactronan when you in high nitrogen environments, and this promotes then the cleavage, and it produces more of these fragments that are actually going to suppress the defense reaction. So this is an example where you and to understand the structure function of your polymer, you need to also take into account that it also plays a role in the indirect interaction with pathogens or other microbes. So this is, I think, a nice example of the utilization of the smart utilization of pectins. It's where they made a temperature sensitive layer. So you can make a film of demethylated pectin here, and you add calcium. So the calcium binds to the negative charges of your pectin. And with this film, they were able to make a artificial skin that can detect in a very sensitive way changes in temperature. And this here is this is in particular striking. They compared it to this pit. What do they call it? Pit membrane of a rattlesnake, which is the rattlesnakes are very good in detecting very small changes in temperature. And using this pectin film, they had this similar sensitivity as the rattlesnake pit membrane. So it's an example where you get a very interesting material out of using the properties of the pectins to be able to bind calcium and release it in changing temperatures, for instance. So I think that's an exciting example. Okay, so now something about the outlook. So what we need to do now in the coming years is to understand what all these polysaccharides are for, but we need also to understand their structure. And so there's a lot of new techniques that are emerging now to characterize polysaccharides. This is the work of the group of Frengel. He's making a lot of chemically synthesized oligos, put it on chips, and you can use that to bind, for instance, antibodies or polysaccharide binding proteins to characterize it. It's a very nice tool. Another thing is to look directly on polysaccharides. This is with scanning tunneling microscopy. You can actually visualize the structure of your oligosaccharide using this technique. So it's a really interesting advance. And Kalina, she's developing in our group, she's developing the super resolution microscopy to do glycopainting. So you can use different probes that recognize demethylated homogalactrona or rhamnogalactrona and backbone is another. You can look, here's, it's maybe not so easy to see, but this is purified pectin, and you see that you can distinguish like the polymer on the that has been deposited on the slide, and you see that you can distinguish also connections between, for instance, RG1 and homogalactrona, which people haven't been able to see so far. And you can also see it in vivo here in a root hair cell wall. You see that, for instance, homogalactrona is making areas that are devoid of, or the areas that are devoid of homogalactrona that are filled actually with rhamnogalactrona, which is really also very exciting. You have that at very high resolution. The problem is that you're limited by the size of the polysaccharide binding probes, which are often antibodies, which are quite big compared to the limits, the resolution limits that you have. So other advances are now to synthesize polysaccharides, for instance, in yeast, but they are confronted with the same problems as Anne was mentioning, that often you don't have the nucleotide sugars in the heterologous host, so you need to introduce those as well, etc. So it often is complicated. But the advantage is that you then can make more homodispersed polymers with defined substitution patterns that are much easier to when you want to do structure function analysis, and you also can produce it in vitro, of course, when you have all the enzymes. And then in vivo function, there's some really nice tools to image cell wall properties using it's Joris Brakel who made these tools. They are based on a boron here that can cross-link sugars in the cell wall, and you can label the cell wall like that. And he was able to put like probes for cell wall pH, porosity, viscosity, ROS levels. You can measure that in the cell wall. So and Kalina showed an example last week. It's used. So to conclude, so the cell wall contributes to the diversity that is relevant for plant adaptation to abiotic and biotic. Should be biotic environments here. The cell wall metabolism may affect greenhouse gas emissions, so we need to take that into account in the evaluation. And also it indirectly can affect the carbon storage, as I give an example. Exploiting cell wall components obviously contributes to the bioeconomy. The perennial grasses can be of interest in certain conditions. Knowledge on polysaccharide structure function is essential to understand plant biology in general, but also can be exploited to make smart materials, for instance, also to selecting feedstocks for the bioeconomy, and connecting polymer physical chemistry to cell wall properties and plant growth requires, I think, give the example that you need to manipulate and observe it in simple systems with very high spatial temporal resolution to be able to make these causal relationships. So I will finish here and thank the people in the group and some of our collaborators that have contributed to things that I mentioned today. And I thank you very much for your attention.